Alrighty, in today's video, we'll be discussing the lungs. So we're just going to talk about the layers of the lungs and the pressures associated with them. So here we have the visceral pleura and the parietal pleura. Now in between these two layers is called the pleural space, or the intrapleural, and then continue on to that, the pleural cavity. And now this is filled with fluid and allows these two membranes to slide against each other because there's liquid filled in between, and it creates surface tension. Now in this area, there's an intrapleural pressure, and it's a negative pressure right here, and it goes between -3 to about -8. And this pressure actually allows the alveoli, and this is a picture of an alveoli, this is a picture of a lung, to expand, and for the alveoli, the smaller portions, to expand and go from this to that. And that's what creates a pressure gradient. Now the pressure outside, we're going to say 0, so then we'll have just a net inflow inside the lungs of air. So hopefully that makes sense. Here's another thing I want to hit on. Increase in volume is going to decrease pressure, okay? And that's vice versa. A decrease in volume will increase pressure. That's something to remember well in this video. We're going to briefly talk about pneumothorax. And now this is basically occurs when the pleural cavity is exposed. So either through like a knife cut, or just maybe a rib punctures it. So air enters pleural cavity. Okay, and if air from the atmosphere enters this cavity, we're going to have a loss of negative pressure. Now with the loss of negative pressure, that means the lungs can't go from this to this, right? They can't expand. And so what's going to happen is that you're actually going to have the lung collapsing. So the lung actually collapses without the negative pressure. Okay, so that's a pneumothorax. Now we're going to talk about, we'll go back to Laplace's law, but this is kind of all intermingled. We're first going to talk about surfactant. So here's an alveoli, this guy here, this whole thing. In the alveoli, there's a cell called the type 2 alveolar cells, or pneumocyte type 2, and these cells release surfactant. And now it's important to remember that surrounding the edges of this alveolus are hydrogen or water, H2O, and that's what it creates surface tension and allows the alveoli to be pulled in for when you're exhaling. It pushes air out. See, now surfactant actually breaks down these hydrogen bonds and creates what's called compliance, and allows the lung to expand and decreases surface tension. And it prevents the alveoli from collapsing. Now we're going to talk about Laplace's law. So Laplace's law basically just tells us the pressure based off of surface tension and the radius of the sphere. I'm going to talk about emphysema really fast. So in emphysema, the alveolus actually gets big, becomes very compliant. And so that means that the radius gets big. Now if the radius gets big, pressure will decrease. And that makes it really easy for air to actually come into the lungs, right? makes it really easy for air to come in. But since it's very compliant, it's like saran wrap. It expands super easily, but if it tries to recoil or go back to its original state, it's extremely difficult. So it's hard to breathe out. Yeah, so that's basically there. Let's see. And then I guess we could say that with smaller alveoli, they would collapse without surfactant, which we just said previously. So hopefully that's just makes sense. I mean, if we have a decrease, we have a decrease in tension or surface tension, we would actually have an increase in pressure, right? Because it would be closing, it would actually be going this way, right? Instead. Okay, so that's that. We're going to talk about another equation, the alveolar gas equation. And this is just talking about the pressure of oxygen in the alveolus. Now, I'm just going to go through a couple different... We're just going to go through the equation. I'll explain each thing. So the FiO2 is a fraction of the inspired oxygen. Okay, usually for us, it's about 0.21 normally. This PATM, that's just atmospheric pressure, so that's around 760. This pH2O is usually constant around 50. It's just diluting the oxygen, right? It's just the pressure of water. Now we're going to get into this portion here. And basically, right now, it's PaCO2 over R, which gives us our O2. But it's how O2 is taken out of alveolus to then be burned or metabolized. Now, if the patient is healthy, these are the values that we're going to see. But, overall, this just helps us explain alveolar oxygen levels. And this PA2 will normally be around like 102. Okay. And it just kind of helps us assess like ventilation, oxygenation, shunting, and then we'll later, actually, we won't talk in this video, but later, the VQ ratio. Now we're going to talk about how CO2 and O2 are exchanged from the lungs into the blood, vice versa. So here in this image, we have an alveoli, and it's next to a capillary. Now, when we breathe in, let's go with this one. When we breathe in, oxygen is going to fill this up, right? So it's going to cross this right here, and it's going to bind to hemoglobin, okay? Once it binds, there's 4 binding sites for O2 on hemoglobin. And once it becomes saturated, it will kind of go away from the blood from this area. Because I think this is the biggest part to know. I'm sorry this image is confusing, but because there's such a high pressure of O2 in this region, that's what allows for hemoglobin to have a greater affinity for O2 than CO2. And so that's why O2 is able to bind to the blood. And then we'll talk about later in the R and T states, but I just want to explain that portion first. And then once we get to the cells, the O2 is actually going to be released, right? Because it needs to go to the tissues. And CO2 is going to be coming from the tissues into the blood because it's a waste product of metabolism. And it's going to come in and it's going to bind to hemoglobin. And it's going to kick off an O2. And this will trigger what's called the T state. And the T state basically just means that O2 is being kicked off of hemoglobin. And then just to briefly talk about kind of where CO2 is found during this exchange. So 7% will be found in the plasma, 23% will be attached to hemoglobin, and then 70% will actually be converted into carbonic acid, HCO3, bicarb, and that's how we'll go through the blood. Yeah, and then I think we'll go back up to here. So now we're in the lungs, and I'll briefly explain just a couple things that happen here. For example, if the person is in acidosis, right, there's a large amount of protons, right, or a ton of CO2, whatever. That's actually going to compete. It's going to do the same thing as CO2. It's actually going to push off the O2 and inhibit O2 from binding to hemoglobin. So then we would actually, this would technically lead to a hypoxic state for this individual. And now, like, other way around, yeah, so that's kind of what I was saying. So that, and then also carbonic anhydrase, and there's another molecule we'll explain later. It's called 2,3-DPG, which basically just also kicks off O2 and triggers it into a T state. And CO2 does that because it's basically an acid, and temperature will do the same thing as well. So here we have this image. This is a graph talking about the oxygen hemoglobin dissociation curve. Sorry, I probably should. This is the O2 saturation percent, and that. Oh man, what was that? I'll figure it out. 1 second. I can't remember the x-axis with that. Oh, that's a PO2. Sorry. PO2. Okay. So a right shift essentially causes more O2 to be released into the tissues. So technically like a T state. Right? So if that occurs, usually what triggers that is a low pH, right, the protons, or high CO2, which eventually actually turns into protons. The temperature increases, or if 2,3-BPG also increases. 2,3-DPG essentially just is able to bind to the hemoglobin, and because it's able to like so 2,3-BPG converts a 3-carbon chain with 2 phosphates, and it rearranges the molecule in a way so that it fits perfectly as such as like a key. It sticks into the hemoglobin, attaches to the hemoglobin, and kicks off the oxygen. So that's what's going to happen there in a right shift. In a left shift, we're actually just going to have the complete opposite. So an increase in pH, so less acidic, decrease in CO2, right? Decrease in temp, decrease in DPG. And this is actually going to increase the uptake of O2 in the lung, or an increase in oxygen saturation. And you'll actually see this in a fetus, because fetuses don't have 2,3-DPG. So in the placenta, 2,3-DPG will actually make it so the red blood cells are in a T state, so releasing the O2, and the fetus will actually have a super high affinity for the O2. So fetus blood is actually has a higher O2 saturation. Yeah, and that's just a quick overview of the lungs. Yeah, if you have any questions, let me know.